Chapter 14, Biotechnology. Thomas Hainsworth spent 27 years living in a waking nightmare. This chapter starts off with he was imprisoned for crimes which he did not commit. 27 years later, 27 years in prison, after which finally he was released due to DNA technology, due to biotechnology, he was released. So this chapter, among other things, will examine how that technology works. Before we begin with a more scientific look at what biotechnology is, let me first give you a comparison. We can consider species, different species, mice, people, plants, like different books. They all have different genes, and those genes are read. The information is used to make that organism, whether it's a mouse, a tree, or a person. So what can we do with the information in books? Well, digitally, it's very easiest for us to, well, first read it, obviously. Secondly, we can copy paste a sentence from one book, put it into the same book. So now there's two copies of that sentence. We can take one sentence in one book and we can very easily just delete it, or we can cut and paste it into another book. And DNA technology uses naturally occurring processes such as enzymes, also the way viruses and bacteria infect their hosts, to essentially do the same thing. And we are approaching the point where we can essentially do with DNA what you or I can do with a word editor. We can write it ourselves from scratch. We can cut and paste. We can take it from take anything from one organism, put it in another organism, and it is becoming increasingly easy and cheap. Even 15 years ago, however long that was, I'm 38, that was when I was 23, I did this stuff as part of the lab. We genetically modified plants, and it's really easy and it's pretty cheap, and now it's even cheaper. So what is it from a scientific perspective? It's obviously not cutting out English sentences from one book and putting them in another. Biotechnology is the manipulation or modification of living organisms or their components to produce useful products or services. So not just DNA, but other stuff, specifically enzymes. That's one of the big ones. So genetic engineering, that is to manipulate cells or organisms by adding, deleting, or modifying genes. Could be for disease, could be for a commercial product, agriculture, you name it. The result is the creation of a genetically modified organism. In recombinant DNA, genes from one species are spliced into the genome of another species, and this creates a transgenic organism. So the mouse here, this mouse has received a gene from a jellyfish which can glow in the dark. So now the mouse, it glows in the dark. Why is its fur not growing? Because the fur is not made of cells. It's an extracellular protein called keratin. But if you were to shave that mouse, then it would be glowing everywhere. All of its skin glows, all of its tissues glow. This, lastly, genetic screening. They don't introduce this in the beginning of the book, but this is a huge one. Genetic screening, essentially, if you give a DNA sample, willingly or unwillingly, I might add, scientists can look at every single gene and allele in your genome very, very easily. And they can test you for all kinds of things and increasingly controversial things. To start off with, obviously they can screen for genetic diseases, but we may be getting to the point where we can screen people for things like proclivity towards violence, manic depression, perhaps even sexual orientation. So all kinds of things are becoming possible, and that of course opens up uh, a lot of ethical questions. Bacterial transformation. So one of the most basic genetic technologies that existed a long time ago, 20, 30, I don't even know how long ago, a long time ago, anyway, is bacterial transformation. And what this involves is the addition of one or more genes into a bacterial chromosome or the bacterial cell. Let me review bacterial structure real quick. Bacteria have a cell wall. Inside of that is a cell membrane. Then they generally will have one single looped piece of DNA called the bacterial chromosome. Sometimes they will have a plasmid, which is essentially a much smaller looped piece of DNA. Bacteria can be caused to absorb DNA from the environment, and it can be just a gene or a chunk of DNA that is randomly in solution near them. It could be an entire plasmid, as is shown here. Could be anything. So we can take advantage of that and put our gene of interest into that bacteria. For example, we want to make this bacteria produce some kind of product of interest. Say somebody is short on growth hormone for some reason, and we need to manufacture growth hormone. So we could get it out of tissue, get it out of human tissue, or maybe another animal's tissue. That's difficult, long, expensive. Or we could insert the gene for growth hormone into a bacteria. So let's say we manage to produce a gene in large amounts. We put that gene in the same solution as this bacteria, and then we do what's called making the bacteria competent. We essentially sort of threaten it. We make it worried, so we can heat it up a little bit, and the bacteria becomes what is called competent, and that means it's competent in absorbing DNA from the environment. It kind of, well, the way you can kind of think about it is the bacteria panics and looks for ways to get new tools to survive. So it just starts absorbing DNA from the environment. And that DNA can be our gene of interest. And so this column of cartoons on the left shows a DNA fragment getting absorbed into a bacterium. And in this case, that gene is integrated into the bacteria's chromosome. So the bacteria itself will use enzymes, DNA ligase to connect fragments, and other enzymes that cut DNA to make a hole in the chromosomes so that the new fragment can be added. Other option is this: we can make a plasmid. Plasmid DNA is a little bit more stable. It's looped. That makes it a little more stable. It doesn't have those ends that can kind of break apart because they're just connected by hydrogen bonds. And we can put many genes into this bacterial plasmid. The advantage of that is that bacterial plasmids can replicate many times. So a single bacterial plasmid may have 10 or 20 copies of the plasmid, and that just means it is more, it can be used more often, so the gene can be transcribed and translated more often, and it also increases the copy number of the gene, which can be useful for other applications. Okay, so two ways that we can transform a bacteria by adding a gene of interest into the bacteria. Viruses can also be useful in moving DNA from one location to another location. First, let's take a look at how some virus life cycles work, or replication cycles, I should say, because they're not technically alive. So number one, a virus will attach to a susceptible host cell. So that's what we can see happening right here in number one. And that is fairly specific, which can be useful because we can get a virus that will only infect certain types of cells. So for example, if we wanted to genetically modify only a person's liver tissue, for example, if we can find a virus that only infects liver tissue, then that virus will only get into the liver tissue and leave everything else alone. We hope. Number two, the virus enters the host cell. Number three, the virus releases its DNA into the host cell. Some viral DNA in red may be incorporated into the host cell's DNA. So if this was a eukaryotic cell or a human cell, we can see the virus has inserted its DNA in red, and that red DNA can be incorporated into the host's DNA. We could also, by other means, insert our gene of interest into the virus, and then allow the virus to replicate in lab conditions and then infect a tissue of interest with that virus. Then the virus inserts our DNA, our gene of interest, into the host's chromosome. Number four, the viral genes encode the synthesis of viral proteins and viral gene replication. Some host cell DNA may attach to the replicated viral DNA. So here is sort of an sometimes uncontrollable outcome of viral infections. That is that when they are copying their DNA that is in the host's DNA, they're not very good at copying their DNA. They make a lot of mistakes. So sometimes they will copy the host's DNA as well. And what that means is that they will reproduce themselves along with some of the host's DNA. So you can see this viral particle that has been reproduced inside of the host. And it has its own red viral DNA, and then it also has that blue DNA right here, that blue DNA right there. And that is some DNA that was just downstream or upstream from the viral DNA. I'll erase that now. And so what that means is that sometimes a host's DNA can be pulled out of the host and into the virus. And then when the virus goes to infect another individual or even another species, that host's DNA can transfer from one species to another species. Some scientists have even expressed concern that as the permafrost melts in the northern parts of Siberia, Canada, etc., that when dead animals that died tens of thousands of years ago thaw out, viruses that infected them may be able to excise themselves out of the host's genome and come back and infect humans, other animals, or other plants, and that those viruses may be just so old that people don't have much resistance to them. How likely is it? I would say that it isn't really that likely, but it is a possibility because a virus can exist. Take a look at number 5 here. I'll circle it. A virus can exist just as genetic code in a host's chromosome, just as a sequence of base pairs. A virus can exist like that. That is how some STDs exist. For example, herpes. Viruses just wait in the host's genome, and they have ways of sensing when the host is weakened. And then when the host is weakened, say you get the flu or something, or somebody who has herpes gets the flu, they may be more likely to have an outbreak because the herpes virus can detect that the body is busy fighting off another disease, and therefore it will burst out of the genome, replicate itself a whole bunch, burst out of the cells, and cause some unpleasant symptoms. So let me conclude by saying this. Remember that we can put our own DNA into viruses pretty easily, and that because viruses are somewhat specific in terms of what tissues and what cells they'll infect, we can use them to deliver genes of interest to various tissue types. CRISPR-Cas9, for this one, I'd like you to check out the video link on Blackboard. It does a great job of explaining what it is and how it works. DNA profiling technique, so looking at what types of alleles an individual has, identifying who was here based on DNA, solving crimes, solving paternity cases, etc. First step is copying the DNA a lot, because typically wherever you find a DNA sample that you're interested in, there is not very much of it, so you have to copy it many times. This is called the polymerase chain reaction, or PCR for short, and it's a technique to quickly and precisely copy a specific segment of DNA. Let me add to that, you can copy just specific regions of DNA if that is what the person is interested in. For example, if a researcher was interested in copying only one gene out of the human chromosome, human genome, they could do that. Could also just copy all of the DNA generally in a sample. So how does it work? Basically this. Let's take a look at generation number 1, or the number of DNA molecules. First, we start off with one DNA molecule. And it's true, this is not an exaggeration. If we find just one molecule of DNA, then we can make millions or hundreds of millions of copies of it quite rapidly. This works through rounds of heating and cooling. So first, the sample is heated, which breaks the hydrogen bonds that link the complementary bases together. Then we get 2 single strands of DNA. We add DNA polymerase, and that DNA polymerase copies complementary strands to each of the parent strands. So then we have 2 molecules of DNA as shown here. Now we have 2. We repeat the heating phase of PCR, and all strands of DNA present will break apart into 2 strands, 2 single strands with their complementary bases exposed. And then we, again, same thing, the enzyme is added and the complementary bases are added by the enzyme. And now we have 1, 2, 3, 4 molecules of DNA. So there's rounds of doubling. So it goes from 1 to 2 to 4, 8, 16, 32, and so forth. So very rapidly, a small amount of DNA can be copied and become a large amount of DNA. PCR is used very commonly because in pretty much every type of genetic engineering, any kind of DNA technology, more than one copy of the DNA is used. We need a large sample. Guilty or innocent? PCR was used to amplify DNA found in a 25-year-old sperm sample. The DNA was analyzed as follows in the next slides. And so this guy, he was proven to be not guilty of raping 5 women. He spent 27 years with the criminal justice system saying that he raped 5 women when he in fact did not. In part, he was shown to be guilty based on eyewitness testimony. Eyewitness testimony has very strong evidence for being not reliable. And so DNA is a much, much better way of determining who was in fact at the scene of the crime, or at least whose DNA was at the scene of the crime. Gel electrophoresis. So this is one way, a pretty simple way, rudimentary way, very kind of an old school way nowadays, that we can determine whose DNA is whose. So let me point out a few things here. On top, this is the crime scene DNA, and then this is the suspect's DNA. So in some manner, the two pieces of DNA are going to be compared. So if the suspect's DNA is the same as the DNA at the crime scene, then we say the DNA at the crime scene must belong to the suspect's DNA. Basically, this is what goes on. We are going to cut up both samples of DNA in specific ways, and the DNA is cut up based on the base sequence of the DNA. For example, let's take a look right here. We could use one enzyme that cuts anytime it finds the sequence CCGG. So it cuts anywhere it finds CCGG, and it cuts in kind of a diagonal pattern, as you can see, based on the red arrow already present there. It'll cut the sugar phosphate backbone. Obviously, the hydrogen bonds will also be broken, and we get 2 fragments. Then it's going to be cut here as well. These are called restriction sites, because the enzymes used to cut the DNA are called restriction enzymes. Where did these enzymes come from? Well, we found them in the natural world, typically in bacteria. Bacteria use them as defense against viruses. So when a virus inserts its DNA into a bacteria, the bacteria's got all of these restriction enzymes that'll break down any DNA that does not belong to the bacteria. All right, so the basics here. Your book goes into a little bit more with the detail, with these short tandem repeats. What do you need to know? They vary in number between different people. So if there's more of them, then you're going to have a bigger chunk of DNA present. If there's less of them, you'll have a smaller chunk. Next, it may also be used in RFLP analysis. That's what I just mentioned right here. Restriction enzyme fragment length polymorphism analysis. Aren't you glad that we don't need to know the details of that? All it means is just how many of these restriction cutting sites are there present in the DNA. So if there's 2 present, then the DNA will be cut twice. So let's take a look at the crime scene DNA in a little more detail now. So there are 2 places where the crime scene DNA is cut. So this one piece of DNA, if we cut it twice, is cut into 1, 2, 3 fragments. Then you can see down here, there are 3 fragments of DNA here as well. Why are they separated in this manner? I'll explain that in a moment. The suspect's DNA only has 1 restriction site, or 1 CCGG sequence, so it is only cut 1 time. It's cut 1 time so that we now have 2 fragments, Z and Y. So the suspect's DNA has a fragment that is Z, and then another one that's Y. So looks like they have 2 fragments that are the same. The crime scene DNA has a Y fragment, and the suspect's DNA also has a Y fragment. And there you have it. There is some similarity, but they are not the same. Let me go back to that enormous word, RFLP, restriction fragment length polymorphism. Polymorphism, that means a change in the DNA. So if you notice right here, suspect's DNA, they have A CGG instead of CCGG. So that's a polymorphism, a single base polymorphism, or a single nucleotide polymorphism, just one mutation. One nucleotide's been changed, and we can compare the 2 samples of DNA by cutting them up. It sounds kind of crude. It is, I guess, pretty simple. The suspect's DNA wasn't cut there because it didn't have a CCGG. It had a ACGG. All right, so Now, coming down to this X, W, Y, all of this right here, I'm going to explain it briefly on this slide, and then I'll go into greater detail on the next slide. Basically this is a gel. So this is called gel electrophoresis. So this gray kind of, I don't know, Lego castle wall looking thing, this would actually be made of jello. We would put it flat inside of a special solution, and then we put the DNA here from one sample and DNA here in another sample. Next, we're going to connect it to electrodes. So this is going to be the negative end. So that's where the electricity is going to go in, and the electricity is going to flow towards the positive end. Remember that the sugar phosphate backbone of DNA is negatively charged because phosphates are negatively charged. So the DNA will be pulled towards the positive end. Positive end's here. This whole end of the gel, by the way, is all positive. So electricity is going through the gel at every point in the gel, from the top to the bottom. Let me erase some of my scribbles now. So what happens is smaller fragments will go farther. So the Y fragment here is the smallest fragment, and it looks like both the crime scene DNA and the suspect's DNA both were cut in such a way as to produce a similar or exactly the same size fragment. And then the suspect's DNA, since it was only cut one time instead of two, had a really big second fragment, so it didn't move very far. So bigger fragments just move less far because they're heavier, and the electric charge that is pulling the DNA downward here is not going to pull them as far. Here is just a more accurate cartoon diagram of gel electrophoresis. So there's a power source, negative, positive. The electricity, again, flows through the gel. I know they've kind of got this diagram here that looks like it's going around it. That's not true. It goes through the gel at every point in the gel, through the gel. This is electricity going through the gel. DNA is pulled towards the positive end due to the negatively charged phosphates, and smaller fragments go farther. So basically here, we at the end here, we've got 3 samples of DNA that they are comparing, and it looks like none of them are the same. And again, here are the largest fragments. Here are the smallest fragments of DNA. DNA profiling. So can be used to determine if 2 samples of genetic material are from the same individual. Here, can you determine which suspect left some DNA at the crime scene? Blood at the crime scene, and suspect 1 or suspect 2. And I imagine that you could use some critical thinking to solve this problem, even if you had no idea what I'm talking about. So look at this. See that? It's similar to this. So those 2 are the same DNA. We can also conclude, or we can conclude from this that both the crime scene DNA and the suspect DNA were cut in the same location, therefore producing the same sized DNA fragments. Suspect number 2, their DNA was cut at different locations, producing DNA fragments of different sizes. Investigating murder, paternity, and ancient DNA. So DNA profiling can be used to test the guilt of suspected criminals. Again, it only determines if their DNA is the DNA that you have found there. So it doesn't mean they necessarily committed the crime, but it does mean that they were there at the crime. They left the blood there, they left the sperm sample there, they left the hair follicle there, whatever the tissue sample is. Identity tissue samples of victims can resolve paternity cases and can trace the evolutionary history of organisms. DNA analysis can also be used to compare the DNA between closely and distantly related species to infer how recently the 2 species being compared had a common ancestor. So looking at this sort of bar chart, I suppose we could call it, what we see here is different colors shown for each different base. So there's a green, a red, a blue, and maybe a black for the 4 bases for each of these 4 species. And then between them, so for example, between the honeybee and the bumblebee, the number of differences is shown right here. So at each bar, so there's a bar, there's a bar, there's a couple more bars. At each of those locations where there's a bar, that indicates a single base pair difference. So that's how many differences there are between those two species, the bumblebee and the honeybee. And then if we look at the American robin and the hermit thrush, again, we see the same thing. This is the number of differences between the two species. Now, if we look at the bumblebee again and the American robin, the number of differences between these two species is significantly more. And that is probably not very surprising because they're much different organisms. Note also that they have probably more similarities than they do differences. So the fact that they are similar in terms of the base sequences, yet there are differences, indicates that they share common ancestry. For example, what kinds of similarities might they have? They're pretty different species, but remember all of these cellular structures, for example, a protein that pumps a certain type of ion into or out of the cell, ATP synthase in their mitochondria, histone proteins that are used to coil DNA. All of those things are going to be really similar between almost all species, in fact. So looking here, this is a much more zoomed out view of evolution, and we can compare each of these groups of species. For example, jawless fish, they're really old, they've been around a long time. Cartilaginous fish like sharks, bony fish, amphibians, mammals, turtles, etc. We can compare them all using DNA analysis, and we can choose just one gene, for example. All of these genes, for example, have the same proteins in the electron transport chain in the mitochondria, and we could just choose one of them and compare them. And when we do that, scientists have done it. Cytochrome C is an example. You don't have to memorize that specific protein, but when we compare them, what we find is that morphologically similar species also tend to have the same DNA sequence for these various genes. For example, crocodiles and birds will be more similar to each other than a crocodile will be to a mammal. And then if we're a little bit in doubt, remember the scientific method, we need repetition. So we would take another gene. For example, maybe the gene that, let's say DNA polymerase, for example, and we could compare the gene that codes for that enzyme and see how similar they are. And basically, we just kind of reconfirm the same thing over and over. How are transgenic organisms made? So recombinant DNA is constructed when scientists combine pieces of DNA from two different sources to form a single DNA molecule. Recombinant DNA technology is widely used in genetic engineering, the direct manipulation of genes for practical purposes. Here, we've got fish that glow in the dark. Isn't that neat? You can go buy them. I'm not sure if they're for sale at our local pet stores here, but they are for sale somewhere. So how do we get a gene from one organism into another organism? Number one, we're going to take a gene of interest and we identify. So whatever this gene is, let's take the glow-in-the-dark fish gene. So that would be a fluorescence gene. Fluorescence gene, excuse me. DNA cloning. So we have to copy the gene. The gene is put into a single-celled organism, which then reproduces. So this is one way of copying a gene. Instead of using PCR, we can use a bacteria that will just copy itself and any DNA in itself. So here's what we would do. We're going to first take a DNA segment, including the gene to be cloned. We cut this gene out. So the blue part, that's the gene we're interested in. We cut it out using, again, restriction enzymes. We cut it out. And then, what does this one say here? The plasmid and the DNA segment containing the desired gene are cut with the same restriction enzyme. I'll get back to that on the next slide, why that's important. Basically, though, we have the gene. We've cut it out. Now we're going to put it into this plasmid. We have cut the plasmid as well. Plasmid used to be a circle. We cut it once. Now it's got a hole in it. So we take our gene of interest, put it into the plasmid, and you can see now we have a recombinant plasmid. Recombinant means that two samples of DNA from two different species have been combined. We then get this plasmid DNA into the bacteria, and the bacteria replicates. And along with replicating itself, the bacteria will also replicate our gene of interest. Okay, so in the written word, what I just said, restriction enzymes are used to cut out the gene of interest and also cut the plasmid DNA. The plasmid is then inserted into the. Okay, so now a more closer look at the use of restriction enzymes. In blue here, let's say that that is the plasmid DNA. We're going to cut it once using a restriction enzyme, and the restriction enzyme cuts in a diagonal manner. So if we cut that blue sample of DNA, it's going to result in what are called sticky ends. So they're called sticky because the ends of the double strand of DNA end in just a single strand of DNA. So we can see the sticky end here is TTAA, and its complementary sticky end on the other side is AATT. They're called sticky because all of them can form hydrogen bonds, and hydrogen bonds are kind of sticky because they can easily form, break, and then reform. If we have our gene of interest in green here that has also been cut with the same restriction enzymes, it will have sticky ends that are complementary to any other sample of DNA that's been cut with that same restriction enzyme. So our gene of interest has AATT as a sticky end here. So it is going to be complementary to one side of the plasmid that's been cut, and its other end will be complementary to the other side of the plasmid. So they will naturally bind to each other by that specific hydrogen bonding of complementary bases. And then we can use DNA ligase to connect the sugar phosphate backbones, making the plasmid whole again. Next, how do we then get it into our target organisms? So what we've done here is I'm giving you an example of we're going to take a gene of interest from a fish. Maybe this fish has an antifreeze protein, like it's an Arctic fish. This is actually a real example. We can take a antifreeze protein from a fish that lives in really cold temperatures. We're going to get that gene of interest, get it into a plasmid, and then the bacteria reproduces a whole bunch. So now we've got a lot of these bacteria. And some bacteria, specifically, you don't have to memorize the name of it. It's called agrobacterium. They are natural genetic engineers, and they will naturally infect plants and insert their plasmid DNA into the plant's genome, take control of it, and then produce what's, well, basically a tumor. And the bacteria live in this tumor and get sugar from the plant because the plant produces sugars by photosynthesis. But if we have put our gene of interest into the plasmid DNA, the bacteria kind of does the work for us. The bacteria will put our gene in that plasmid into the plant. So this is called transfection, kind of a combination of transfer and infection because the bacteria is literally infecting the plant. And what it is a vector to transfer the gene to another organism is selected. So in this case, we are using a bacterial vector to get the gene of interest into our target organism. 3, viruses can also be used as a vector. How would that be done? I already introduced this idea a bit, but basically we are going to put our gene of interest into the virus, and then this virus is going to infect whatever tissue it is we're interested in getting our gene of interest into. So the virus binds to the host and then inserts its genetic material along with our gene of interest into the host cell. And then the virus just does what it does and gets that DNA into the host cell's nucleus and hopefully gets it integrated into the one of the host's chromosomes. The rest of this is not pertinent to this slide. This is just a picture of viral replication. 4, 5, and 6. So number 4, lipid vesicles. These are really not that common. So vesicles can naturally merge with cell membranes, and whatever is inside the vesicle will then be inside of the cell membrane. This is kind of sloppy because once the DNA is in the cell, it's just in the cytoplasm, so it will not necessarily be taken into the nucleus. Various tagging mechanisms can be used to try to encourage that DNA to be absorbed into the nucleus, but it's not a very common means of transfection. Pipettes, this is a little more common, and that is what is shown right here. This is a very, very small micropipette made of glass, and they've got special techniques for making them this small. This thing over here is a very gentle suction device, and it will suck this egg onto the surface of it, and all that does is hold the egg cell still. One of the more famous uses of this technology is to clone an entire organism. So if we get an egg cell and then use this pipette here to suck the nucleus out of that egg cell, then this egg cell has no nucleus. Then what scientists can do, if they are interested in doing it, is they can take a nucleus from another individual of that same species. For example, a sheep. Dolly the sheep was the first to be cloned. So these scientists took a nucleus from Dolly the sheep and then put it in another egg cell of another individual sheep. And so basically they just transferred the nucleus of one sheep into an egg cell of another sheep. Then they put the egg cell into the uterus of a different sheep. So then it implanted and came to full term and the sheep gave birth. Also, just smaller pieces of DNA can be inserted in this manner as well. 6. Great care must be taken not to insert the gene in a location that disrupts other genes. That is particularly true with viruses. Viruses do not particularly care about their hosts, so they will insert their genes anywhere in the host. Different viruses are more and less predictable in terms of where they will insert their genes. And by where, we really mean what sequence of bases will the virus bind to and insert its DNA into. It's, like I said, predictable with some viruses and very unpredictable with other viruses. And that's a reason why genetic modification can sometimes cause cancer. These viruses might insert our gene of interest or maybe some of their genes right into the middle of an oncogene, or a proto-oncogene rather, thereby disrupting one or more genes that regulate the cell cycle. The cell cycle becomes unregulated. Cancer can be the result. How are genetically modified organisms used? They're being used to feed people, basically, i.e. increase crop yield, decreased by decreasing losses due to pests and other mechanisms. And the food that contains genetically modified organisms that we eat is not at present required to be labeled. 2. 90% of soybeans, corn, and cotton in the U.S. is transgenic, and many other crops are also genetically modified, although to a lesser extent in terms of the percent of total crops grown. One way that crops are engineered to increase crop yield is by insect resistance. The example here is Bt maize. Bt stands for Bacillus thuringiensis. That is a kind of bacterium. So bacterial maize, basically, i.e. a gene from a bacterium has been put into maize or corn. So Bt maize has been genetically modified to express genes and proteins from Bacillus thuringiensis, which are poisonous to some insects. The protein is expressed in all cells of the plant, and so when an insect eats the plant, the Bt toxin kills the insect. Why is the protein expressed in all cells of the plant? Well, that's simply because they put this gene into the zygote of the plant, and the zygote, that cell, divides by mitosis, which produces genetically identical daughter cells. So by engineering the zygote that produces the adult plant, every single cell in the plant is going to have this gene, which will be expressed, in this case, as a protein. So every tissue type, every cell type has a compound in it that is poisonous to insects. It is useful because there are different strains of this bacterium which produce slightly different poisons. Different strains of B. thuringiensis produce different toxins which can be inserted into the maize to protect it from different pests. Also, the pests are more or less guaranteed to eventually evolve resistance to one or more of these toxins. So different types of toxins can be engineered into the plant as needed. Down here we can see that's a real picture of a corn borer. It's from Europe, and that was the main reason that scientists became interested in engineering corn to kill this insect. And here, this is just a picture of the video that you can access on Blackboard to look at genetically modified organisms. Detrimental impact, a damaging effect on other species of insects. So are we interested in getting rid of all the monarch butterflies? Well, if we are, we're doing a good job of it. The pollen from Bt maize disperses up to 60 meters, that means about 60 yards, and expresses the Bt toxin. So even the pollen produced by these plants has, has that poison in it. Half the monarch butterfly population in the US feeds nearby maize, most of which is Bt maize. They did an experiment, and 0% of the caterpillars feeding on non-Bt pollen-dusted flowers were dead after 4 days. So what they did was they took genetically modified corn and its pollen. They put it on other flowers to kind of simulate pollen dispersing from the corn in the wind and landing elsewhere. And then they let butterflies feed on it. And not surprisingly, none of the caterpillars that will later give rise to the butterfly died. But when the caterpillars that give rise to the butterfly are fed Bt pollen from that corn, about 50% of the caterpillars were dead after just 4 days. So we are spreading this toxin around in the environment in a more or less uncontrolled way because it can be spread by the wind. Another detrimental impact is that it gets into the water. So leaves and pollen from Bt maize are washed into streams. The larvae feeding on the leaves have been shown to also have a reduced growth rate. And you're probably thinking, well, I'm not really that interested in larvae. Well, you might be interested in fishing, though, and fish eat a lot of these little grubs. Also birds eat grubs, and so there are potential effects all the way up the food chain of producing this poison that kills insects. Also note, insects are more or less the basis or the bottom of every food chain on the planet. So engineering a crop that is grown across a huge amount of the surface area of the United States that kills insects might not be a good idea, but it is also more complex than that because the poison does break down eventually in the environment also. So once it's there, it's not like it stays there forever. It can also be broken down in the environment. This is kind of an interesting example of one town's gratitude towards this pest. You can see that big black, it's called a snout-nosed beetle, or a weevil rather, and they erected a monument to it. Here's a real picture of black and white a long time ago. So the Boll Weevil Monument, December 11th, 1919, in profound appreciation of the boll weevil and what it has done as the herald of prosperity. This monument was erected by the citizens of Enterprise, Coffee County, Alabama. Why on earth did they erect a monument to an insect that destroyed the agriculture industry of their area? Here's why. So they erected it in 1919 and because they perceived that their economy had been overly dependent on cotton, which the boll weevil had just totally destroyed the cotton industry there because it eats cotton. So what the result was is they stopped just farming cotton only, and they diversified their economy and in terms of what crops they grew. And if an area grows a lot of different crops, it's a lot less likely that one pest is going to come by and that can eat all of their crops. So if you have a diverse farm, it's almost impossible that one pest is going to eat all of them. Pretty much impossible. So as a result of their more diverse farming methods, Enterprise, Alabama avoided the worst effects of the Great Depression in the late 30s and also early 40s. Because they diversified their economy in terms of agriculture. Genetically modified animals. So far, this is the only one that has passed all of the various agencies and gotten all the permissions to be let out, and that's the Atlantic salmon. It has an extra copy of the growth hormone gene, so it produces more growth hormone, grows faster. And you can see right here, that's the genetically modified one, and this is the natural one, not genetically modified. Firstly, why might this be good? Well, obviously you just produce more meat, right? It is also going to reduce the amount, if this is in a fish farm, it would reduce the amount of fresh water required. It's going to reduce the amount of feed required, and so we will be able to produce more food to feed more people. Another example, H5N1 resistant chickens. Chickens will sometimes transmit the flu to people, And so if we can make chickens that are resistant to the flu, then they'll be less likely to get sick and then less likely to give the flu to people. And I guess that's simply more or less undesirable than genetically modifying human beings. So it's okay to genetically modify chickens to be resistant to the flu, but not people. I guess that's the reasoning there. Goats that secrete spider silk proteins in their milk. Obviously, the goal here is to produce goats with powers of Spider-Man. Just kidding. That's not it. But spider silk is extremely strong and can be used in various commercial applications. So if we can get goats secreting it in their milk, then we can harvest the spider silk out of their milk. Pretty wild. Also, mice that have human genetic diseases such as Alzheimer's, if we find genes or alleles that are linked to Alzheimer's disease, other ones like cystic fibrosis, the list goes on. We can then study human diseases in mice because it's much more ethically acceptable to do these studies on mice as opposed to doing these studies on human beings. Environmental bioengineering. So just a quick summary of all these things. Number one, rare trees may be engineered for disease resistance. So if a tree species is on the brink of extinction due to a disease, we may be able to engineer that tree to be resistant to it. Bacteria may be engineered to clean up pollution such as oil spills and other toxic chemicals. 3, mosquitoes may be engineered to halt the spread of disease. For example, malaria, which kills a huge number of people every year. Next, how is biotechnology used in medicine? Genetic screening is probably the most important one, and we all maybe know who she is. That's Angelina Jolie. And in the genetic screening world, she became somewhat famous, or maybe the word is infamous, because she had a double mastectomy at age 25 after testing positive for the BRCA1 allele. And that dramatically increases a person's risk of breast cancer. BR stands for breast, CA for cancer. And why do we have one? Well, because there's a number 2 also. There's a BRCA1 and also BRCA2. People who have both of them are at an extremely high risk for getting breast cancer and also ovarian cancer. Genetic screening can also be used to test for any known genetic disease, as well as the presence of pathogens in human tissues. What is genetic screening in terms of a general application? It's a technology that can screen for specific sequences of base pairs. So any application where we're interested in knowing what sequence of bases there are, this can be used for. People who might want to consider using this technology, adults who have a family history of cancer. These adults may want to be tested for the BRCA1 allele. If they test positive, they may want to undergo an elective mastectomy or breast removal. Transgenic organisms produce medicines also. So microbes, generally bacteria or yeast. Why yeast? Because we've done a lot of studies on yeast, so we really understand how it works. That's why yeast. Yeast are eukaryotic organisms that their cells are a little bit more similar to human cells, which can be useful sometimes, but bacteria are good too. Microbes have been used to produce anti-malarial drugs, growth hormone, clotting factors, insulin, and more. So we use yeast to do this sometimes, i.e. we'll take the gene for maybe growth hormone in people or clotting factors in people or human insulin, and we'll put it into yeast. And yeast, remember, they are eukaryotic, so that means they have a rough ER, they have a Golgi apparatus. So that means they can modify proteins in a way that is similar to how a human cell would modify those proteins, and they can be used to produce mature proteins like growth hormone, that's a protein, clotting factors, and insulin. Insulin is also a protein. Otherwise, many of these drugs have to be isolated from plants. Not these drugs specifically. These would have to be isolated. Well, for example, insulin used to be isolated from pig and cow pancreases, which is expensive, and you can't make as much of it because there's only so many spare pig and cow pancreases coming out of slaughterhouses. Other medicines, such as anti-malarial drugs, most of them come from plants. In fact, most drugs that we find come from plants discovered in often the rainforests of the world, just because there's more different types of plants there. Another good reason to not cut them down. So let me conclude by saying transgenic organisms, when we find these plants that have compounds of medicinal use, we can take the gene for those compounds, put it in a bacteria or a yeast cell, and then that bacteria or yeast cell will manufacture that compound like crazy. And that's just a lot easier than actually like planting these plants that grow in the Amazon maybe, and maybe they don't grow here because the weather is different, but a bacteria will grow anywhere. So it's a much easier way to get our hands on huge amounts of these compounds. Gene editing for age. So for HIV to infect cells, it must bind the CCR5 receptor. That is a protein that is on the cell surface of certain cell types in the human body. Stem cells, which produce white blood cells, that's one of the main kinds of cells that is affected by HIV. Stem cells which produce these white blood cells, they can be taken from patients. So we take some of their stem cells, and then the CCR5 gene can be knocked out. Therefore, the white blood cells of these treated patients will not have a receptor for the virus. So when these stem cells that produce white blood cells that have no receptor for the virus are put back into the patient will be more or less cured. The book says that their symptoms will be alleviated. In some cases, they are 100% cured. And the reason for there being some variation in the effectiveness of this is that there are actually some other cell types that HIV can infect. Some cells of the brain, HIV can get into them as well. Ethical concerns and potential benefits. So genetic screening in embryos and adults. Well, if we don't regulate in any way the genetic screening of embryos, we can essentially screen for every known human allele, from genetic diseases, increasingly even to behavior types such as aggression, likelihood of being depressed, likelihood of committing violent crimes, even sexual identity. All kinds of things are becoming possible to screen for. So when we say, sure, we can just screen any embryo, you can go into a hospital and give them a certain amount of money. Maybe it's paid for by insurance, who knows? And we can screen those embryos. And at present, the only type of screening that is allowed is for genetic diseases. There was, I believe, a firm in California that was eventually shut down that was allowing people to select embryos for various eye colors. Lastly, number 3 here, CRISPR-Cas9, that could definitely be used to change human embryos. And if you haven't seen this movie, Gattaca, well, it is a pretty realistic depiction of where we may be headed in terms of genetic screening. So basically, if you want to interview for a job in this movie, they just take a DNA sample and say, well, you're the kind of person we're looking for because you have all these alleles that we think are useful. Let's see. Last thing about genetic screening. Yeah, that's it. Ethical concerns and potential benefits. So, bullet 1, we can certainly increase crop yield. We can produce cheaper fuel. In agriculture, we can also decrease the use of pesticides, and which kill plants, which kill insects. Allergies, there is a concern that, for example, if you are allergic to peanuts and some genetic modification company, they take a peanut gene and put it into celery, then you eat the celery and you have an allergic reaction to the peanut gene in the celery. Yes, theoretically possible. However, it has never been proven. Bullet 3, pollen from insecticide producing gene may be transferred to the wild by insects. And what do we mean by the wild? We mean wild relatives, because most crop species, well, all crop species, have to some extent some wild relatives, and so it's possible that our genetically modified crops can pollinate their wild relatives, thereby introducing a gene that we've put in our crops into the wild. Also, viruses can transfer genes from one organism to another, as stated earlier in this lecture. So if a virus gets into, say, a fish, the genetically modified Atlantic salmon that's got 2 copies of the growth hormone, it could potentially get one of those copies of the growth hormone and insert it into a closely related fish species. Same thing with corn. A virus could get into the corn that has the Bt gene that kills insects, take that gene, put it into another closely related species. Or bacteria, those can also transfer genes, could also produce weeds that kill insects that feed on them. So for example, if bacteria gets a gene out of this Bt corn that kills insects, the bacteria takes that gene, puts it into another species, such as a grass, and then all of a sudden we've got grass growing that kills insects. We might find that to be, well, that's great, but it also could be a problem. We could produce super weeds in the wild that just never have whose population isn't going to be naturally controlled as it otherwise would be by insects, and it could become kind of a problem. Super competitive weeds, what I just said, they might outcompete other wild relatives because they kill insects. This could affect food chains, and biomagnification is where a chemical at the bottom of the food chain can become more and more concentrated. For example, we've got this grass that is producing this Bt toxin, and then an insect eats the grass, and then it becomes more concentrated in the insect because the insect accumulates all the poison from the grass. And then a bird eats the insect, and then it's really concentrated in the bird, and then all of a sudden the bird dies because it's got so much poison in it. This happened with bald eagles maybe 20 years ago with DDT. DDT was undergoing biomagnification, and it accumulated in eagles' eggshells, making them too weak. And so when the eagle sat on the egg, it would just crush the egg. So risk of pollen transfer by wind and insects. So the problem is that many people do not want to eat a genetically modified food. And so some farms grow crops that are not genetically modified. So a small family farm. For example, a large corporate farm, just a little note here that large corporate farms, they have a lot more money, and they do donate money to politicians, and they have a lot more money, so they can donate a lot more money to politicians. They start growing this genetically modified crop. For example, this would be rape, which is used to produce canola oil. And then a bee transfer that pollen, can go up to 4,000 meters. That's something like maybe 3 miles or so. And if that pollen pollinates a local family's farm that is not genetically modified, and the corporation discovers it, the corporation can sue that small family farm for stealing their technology. And this has happened, and it is sad. Large corporation sues a small farm goes out of business. Happened over and over. A couple of different types of farming. Those farms that typically have genetically modified crops, they grow monocultures, and this is what it looks like. One crop for a huge covering a huge area. Excuse me. They're really large, and there are few employees. And what we've seen over the last 30, 40, 50 years is people leaving these rural areas, and these rural communities are falling apart because there's just few employees that are required to do the work for these farms. This farm over here, this, as you can see, there's many different crops here. So a couple of kinds of lettuce. I'm not sure what all those crops are, but these types of farms require many employees. This is called a polyculture, and they are more resistant to pests simply because no pest can eat every crop. One pest might eat this crop here, yum, 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 ate it, but then it doesn't eat the remaining crops, so the business survives. And in these communities, they are more resilient because more people have a job, but then on the downside, the food is more expensive because there are more employees. Adoption of genetically engineered crops in the United States. You can see that we are using more and more genetically modified crops, and that is because the yield goes up, at least per acre, and therefore the food produced by these crops becomes cheaper. Which countries grow the most transgenic crops? Well, you can see the United States is well in the lead compared to every other country on Earth. And you can see that as a good thing or a bad thing. I'll leave that to you. Countries with genetically modified organism food labeling laws. Well, in red, these countries, Saudi Arabia there, and then New Zealand and Australia down there, they require mandatory labeling for nearly all GMO foods, and in the US, not really. For some genetically modified organism foods, but there's no GMO threshold defined. So what qualifies as food that has genetically modified organisms in it? Nope, it's a mystery. Never know if you're eating it in the US. Oregon mandatory labeling of GMOs initiative, Measure 92. This was in 2014. The Oregon mandatory labeling of GMOs initiative, Measure 92, was on the November 4th, 2014 statewide ballot in Oregon as an initiated state statute, where it was narrowly defeated. This measure would have mandated the labeling of certain foodstuffs that were produced with or contained genetically modified organisms. The measure was sponsored by the group Oregon GMO Right to Know. So below you can see what the tally was. So the vote for no was 753,000. So and the vote for yes was 752,737. So it was really narrowly defeated. Just about 800 more people said no, let's not have labeling. But quite a few people did want it to be labeled. And that is the end of this slide. I thought there was one more, but that's it. So I hope you enjoyed the course, and remember to put in your review of the course, whether you thought it was the best course you've ever taken, just a so-so one, or a terrible one. Please do review the course. Thank you, and good luck on your last exam.